Good evening once again. We are going to have a biology session now. We're going to look at transcription. Last time we looked at replication. Yeah, so we are following what we call the central dogma of molecular biology. That's what we are following. We're going to move from replication of DNA to transcription, then finally to translation. That is what you call the central dogma of molecular biology. Because of time, let us start just now. I'm going to share the screen again for the slides of biology. Okay, transcription. Are you able to see my screen? Can I just have some feedback from someone? Hello? Are you able to see my screen? Can someone just respond so that we progress?
Sure, so we are.
Oh, excellent. So we're going to start transcription. Last time we looked at replication, where DNA was actually making another copy of itself. Okay, so now today we're going to look at transcription. Okay, so what is transcription? On the overview, transcription is simply the process of RNA synthesis under the direction of DNA. So you should take note of the fact that there are different types of RNA that are actually produced. You can have messenger RNA as mRNA, tRNA, which is transfer RNA, and you can have what you call ribosomal RNA. Okay. So it's a process in which DNA is copied to make a complementary strand of RNA. While DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and is single-stranded, while DNA is double-stranded. So what is happening is you're actually making a complementary strand of RNA, okay? And this RNA, you get to produce what you call messenger RNA or mRNA from DNA. Now the messenger RNA is called messenger because it contains genetic information from DNA. It contains genetic information from DNA. Okay, so RNA and DNA use complementary coding where base pairs match up, similar to how strands of DNA binds to form the double helix. Okay. RNA polymerase mediates this synthesis. It mediates the synthesis of an RNA strand in a 5' to 3' direction that complements the DNA strand. Okay, so transcription is one of the stages of gene expression. Remember, when they say gene expressions, they mean the genes actually coming out to express the information or the contents that they carry within themselves. So transcription is part of that stage. Okay. So this diagram here, where this DNA, RNA, and protein is simply showing us what we call the central dogma of molecular biology, where you have DNA replication that is going to result in the formation of messenger RNA. The messenger RNA will undergo translation to produce a protein, a functional protein. So therefore, it makes a lot of sense from the definition of a gene that when you define what a gene is, it is simply a short segment of a DNA molecule that codes for a functional protein, or rather that carries genetic information that can be converted into a functional protein. So it makes a lot of sense when you understand the central dogma of molecular biology. Okay. So we are basically going to look at transcription and the different stages of transcription. So the stages of transcription are three. So there are three main stages of transcription. That is initiation, number one. Number two, elongation. Then number three, termination. So let us start with initiation. So transcription is catalyzed by an enzyme, RNA polymerase, which attaches and moves along DNA molecule until it recognizes a promoter sequence. So this enzyme RNA polymerase moves along DNA molecule until it recognizes what we call a promoter sequence. Now, a promoter sequence indicates the starting point of transcription. So not until the promoter sequence is found, transcription will not be initiated. So immediately you get the promoter sequence, then that will be the starting point of transcription. So a promoter is simply a DNA sequence where RNA polymerase attaches and initiates transcription. So now let us talk about the promoter sequence. In eukaryotes, that is in human beings, animals, in plants, okay? Those are eukaryotes, okay? Unlike the prokaryotes, that's bacteria mainly. Okay, in eukaryotes, transcription factors that are actually proteins that control the rate of transcription also bind to promoter sequences with RNA polymerase. So once bound to promoter sequence, RNA polymerase unwinds a portion of DNA double helix. Remember that DNA is a double helix. So just a portion of it will actually be unwound, exposing the bases on each of the two DNA strands. Okay, the region of unwinding is called a transcription bubble. The region of unwinding is called the transcription bubble. So this diagram is actually showing us the direction of transcription, which is downstream, and the opposite direction is upstream. Okay, so they are showing us the template strand and the coding strand, the template strand and the coding strand. So let us look at what we call promoter clearance. RNA polymerase must clear promoter once first bond has been synthesized. So the promoter signals which DNA strand is transcribed and the direction of transcription. Remember, there are two DNA strands, so you only need to utilize one or use one. Okay, so the promoter will signal which DNA strands to be used so that RNA polymerase can work and form messenger RNA. That was initiation, then we go to elongation. Okay, so elongation, one DNA strand or template strand is actually read in the 3' to 5' direction, and so provides what we call a template for a new messenger RNA molecule. So a template is like what we are going to use as the original to make another copy. So the other strand is called the coding strand because the base sequence of the new mRNA is identical to it, except for replacement of thymine with uracil. Remember that uracil is found in RNA, while thymine is only found in DNA. So take note of of that difference. Incoming ribonucleotides are used by RNA polymerase to form messenger RNA strand. It does this using complementary base pairing. Remember, we talked about the purines and the pyrimidines. We said that cytosine bonds with guanine while adenine with thymine. Okay, so the complementary base pairing principle is going to be used. Okay, and they're going to have all the different bases. So RNA polymerase will then catalyze formation of what we call the phosphodiester bonds between the adjacent ribonucleotides, and bases can only be added to the 3' end. So the strand elongates. So the strand elongates in a 5' to 3' direction. So the strand elongates in a 5' to 3' direction. Okay, so this diagram is actually representing or showing the direction of transcription. Okay, they're saying that transcription progresses at the rate of about 40 nucleotides per second. Okay, in prokaryotes, RNA polymerase is a holoenzyme, which simply means that biochemically active compound that comprises of an enzyme and its coenzyme. It consists of a number of subunits, including a sigma factor or transcription factor that recognizes the promoter. In eukaryotes, there are 3 RNA polymerases, that is RNA polymerase 1, 2, and 3. Elongation in prokaryotes, that was elongation in eukaryotes, but in prokaryotes, elongation begins with release of the delta subunit from the polymerase, and this allows the coenzyme to proceed along DNA templates, synthesizing messenger RNA in a 5' to 3' direction. As elongation proceeds, DNA is continuously unwound ahead of coenzyme and rewound behind it. Then the last stage is what we call termination. Okay, termination, that's the last stage. Elongation continues until RNA polymerase encounters what we call a stop sequence. Remember, it's been elongated, but until it encounters what you call a stop sequence, or elongation stop. So at this point, transcription will stop and RNA polymerase will release the DNA template. So now this will result in release of the newly synthesized messenger RNA from the elongation complex. In eukaryotes, termination involves cleavage of a transcript followed by polyadenylation. Polyadenylation is simply the addition of adenylate or adenine, let me say adenine, sorry, addition of adenine and nitrogenous base, okay, to the messenger RNA, that is polyadenylation. In polyadenylation, a series of adenine residues or a poly A tail is added to the new 3' end of messenger RNA strand. Okay, then in eukaryotes, still RNA polymerase 2 transcribes sequence on DNA called polyadenylation signal sequence, which codes for polyadenylation signal AAA, AAU, AAA in the pre-messenger RNA. Then about 10 to 35 nucleotides downstream from the AAU, AAA signal, proteins associated with the growing RNA transcript cut it free from the polymerase, releasing pre-messenger RNA. Okay, so this diagram just illustrating the termination in eukaryotes, which we've already explained. While in prokaryotes, termination occurs as follows. In bacteria, transcribed terminator or RNA sequence functions as termination signal, causing polymerase to detach from DNA and release that transcript. Then I should get to understand that there are actually 2 ways in which transcription is terminated in prokaryotes. We've got what we call the row-dependent termination and the row-independent termination. The row-dependent termination, you have a protein factor called the row protein, okay? that it depends on. And this is the one that is responsible for disrupting the complex involving template strand, RNA polymerase, and RNA molecule. Then you can have the termination or stoppage of the process of transcription. Then the other mechanism is the row independent termination. Row independent termination, this one does not depend on the raw protein. So in this case, you've got a loop that forms at the end of RNA molecule, causing it to detach itself, In row dependent termination, we have raw protein that attacks along behind polymerase on the growing mRNA chain and near the end. Then the polymerase encounters a run of G nucleotides on DNA template, and it stores. As a result, the raw protein collides with the polymerase. So in the process, the function or work of the enzyme RNA polymerase will be terminated by the raw protein, and transcription of messenger RNA will actually be terminated. Interaction with raw will eventually release the messenger RNA from the transcription bubble. Then in the raw independent termination, this one is controlled specifically by specific sequences in DNA template strand, and as polymerase nears the end of the gene being transcribed, it encounters the region which is rich in cytosine and guanine nucleotides. So messenger RNA folds back on itself, and complementary cytosine G and guanine nucleotides bind together, and this results in a stable hairpin that causes polymerase to stall as soon as it begins to transcribe a region in adenine and thymine nucleotides. So these are diagrams indicating how termination actually occurs. So these are different diagrams. Okay. Then when transcription is done, okay, when transcription is done, we have what we call post-translational mRNA processing. Post-translational mRNA processing, what they mean is, after synthesis of messenger RNA, the messenger RNA that have actually been formed are supposed to be processed, before they are actually used. So how is that processing done? So first of all, you've got what you call capping. So the 5' capping, this describes the addition of methylated guanine cap to the 5' end of messenger RNA. Remember, messenger RNA has got the 3' end and 5' end. So now to the 5' end, you keep on adding guanine. So that is methylated guanine cap added to the 5' end of messenger RNA. That is post-translational mRNA processing. Then also its presence is actually vital for recognition of molecule by ribosomes and to protect a mature molecule from degradation. Now remember that the RNases will actually degrade messenger RNA. Now to protect it from degradation, you actually have the addition of the methylated guanine at the end of the 5' end of messenger RNA. Okay. So remember that recognition of the molecule by the ribosomes, if you guys remember the function of ribosomes, you know that ribosomes, that's where protein synthesis takes place. Now protein synthesis is what you call a translation. So, and remember that translation comes from messenger RNA. The messenger RNA will have to pass through the ribosomal subunit found in the ribosomes. So the ribosomes where protein synthesis is taking place, you need messenger RNA there. So the ribosomes need to recognize the messenger RNA. So it is important that it undergoes this modification or post-translational processing so that it can be recognized by these ribosomes, which are responsible for the synthesis of proteins. Okay, so there are two processes: addition of methylated guanine, then you've got polyadenylation. Polyadenylation, that is the addition of adenine to the 3' end of messenger RNA. So now you see that to the 5' end, you add methylated guanine. Then to the 3' end, you add adenine. Okay, so you're going to add adenosine monophosphates to the 3' end, and you're going to call that polyadenylation of the messenger RNA, the 3' end of messenger RNA. So this is going to stabilize the RNA that you have actually produced. Okay, after stabilizing it, then it can easily go to the protein synthesis machinery, which is the ribosomes. Then there's another process known as pretranslational mRNA processing, which is splicing. Now, splicing involves the removal of what you call the introns. Now, remember that the introns are the non-coding sequences. Okay. Now, what they mean by non-coding sequences, these sections can actually not be converted to a protein. They cannot code for any amino acid. Okay, they cannot code for any amino acid, and they can therefore not synthesize any protein. So there are certain regions within messenger RNA that are designated for protein synthesis, while others are not designated for protein synthesis. So the ones that actually are not designated, or the non-coding areas, you call them introns. Then the coding sequences are the extrons. So now, removal of the introns or the non-coding sequence via spliceosome, which is the enzyme complexes. Then the joining of the exons, so you've got introns and exons, which are the coding sequence, which are then joined by ligation. So this is a sequence dependent and occurs within the transcript. So you want a situation where you don't want to take messenger RNA to the ribosomal protein synthesis machinery, messenger RNA, which is actually having certain regions which cannot code for protein synthesis. So you want to remove those parts that cannot code for a specific amino acid by using the process or enzymes known as spliceosomes that will cut them off, then later on join the pieces that can code for amino acids or for proteins, okay, by ligation, okay. So that you can have a proper messenger RNA that conserves the genetic material, and can therefore be used to code for functional proteins. So if you see this diagram, which is here, it is actually indicating what is happening, which one is the exon and which one is the intron. So the exon is the coding region. So you're supposed to understand this because there are some MCQ questions that are actually designed just here. You guys would understand what exons do and what introns do. So the exons are the coding regions, then the introns are the non-coding regions because they are non-coding. They are therefore with the coding ligated. Then have you seen on the 5' cap, there is addition of the methylated guanine. Then on the 3' end, there is addition of adenosine triphosphate, forming the poly A tail. So that is post-transcription modification. So by the end of transcription, mature mRNA is made and acts as messaging system to allow protein synthesis. And then within the mature messenger RNA, this one is therefore an open reading frame, which is the region that is translated into proteins. So it is translated in blocks of 3 nucleotides known as codons. 3 nucleotides known as codons. Now those codons, they actually code for a specific amino acid. You are going to sooner or later understand the genetic code so that you see how those codons code for a specific amino acids later on in the sessions. Okay. So at the 5' and 3' end, there are also untranslated regions which are not translated during protein synthesis. Okay. So then we've got a few differences that you should take note of between transcription that occurs in prokaryotes and eukaryotes. You should take note of the differences. Let us take note of finally the differences between transcription in prokaryotes and eukaryotes. So the differences that exist as follows: in prokaryotes, transcription occurs in the cytoplasm. It is important to take note of that. Remember that prokaryotes have got organelles which are not membrane bound, while eukaryotes, their organelles are membrane bound. So that's the reason why in prokaryotes you see that transcription occurs in the cytoplasm, while in eukaryotes transcription occurs in the nucleus. Messenger RNA, once produced from the nucleus, then it is pushed to the cytoplasm for translation. Pushed to the cytoplasm for translation. Then in prokaryotes, DNA is much more accessible. Remember, this DNA is actually outside. It's just floating, free floating in the cytoplasm in prokaryotes. Okay, but in eukaryotes, DNA is actually less accessible to RNA polymerase. Okay, because DNA is actually wrapped around the histones to form nucleosomes and packed to form chromatin, which are other proteins that mediate interaction between RNA polymerase and DNA. So RNA polymerase can actually not just easily get access to eukaryotic DNA because of this kind of protection which is there. However, in prokaryotes, we've got RNA polymerase that directly interacts with the DNA. That is the difference. So it simply means that in prokaryotes, the actual process is less complex. The actual process of transcription is less complex, while in eukaryotes is actually complex. mRNA produced is not modified in prokaryotes, while in eukaryotes, the mRNA is modified by those splicing methods, 5' end capping and addition of poly A tail to the 3' end. So there are actually questions that come. You guys should understand where you add the poly A tail. It is to the 3' end, and the methylated guanine is to the 5' end. Okay. Then the other difference is that DNA is transcribed by one RNA polymerase molecule without assistance of transcription factors in prokaryotes. Okay. While in eukaryotes, transcription factors are needed. Okay. Then in eukaryotes, we are saying you need transcription factors. Then transcription factors are simply proteins that bind to DNA in the nucleus and affect transcription, or have regions that enable them to bind to promoter sequences. Okay, some are called repressors. They bind to regions called silencers that depress the rate of transcription. Okay, in eukaryotes, transcription factors stimulate transcription by binding to a DNA promoter sequence. This stimulates start of transcription. And you've got other transcription factors that bind to regions called enhancer sequences. While in prokaryotes, you've got the RNA polymerase subunit, which has got unique role. The 2 alpha subunits are necessary to assemble polymerase on DNA. Beta subunits bind to ribonucleosides triphosphates that will become part of the nascent, recently formed mRNA molecule. Then we've got the beta prime subunit that binds DNA template strand, and the delta that actually is involved only in transcription initiation. Okay. So those were the differences, and the last difference that you guys should understand is this one. And they actually asked some questions. I remember some questions being asked around this last point. So prokaryotic promoters and eukaryotic promoters. So you should understand the difference between the promoters in prokaryotes and promoters in eukaryotes. So in promoters, at 10 to 35 regions upstream of initiation site, okay, there are two. At 10 and 35 regions upstream of the initiation site, that's where you can find the promoter consensus sequences that are similar across all promoters, okay, and across the various bacterial species. 10 consensus sequence called 10 regions of Pribno box and usually consist of 6 nucleotides. Okay, 6 nucleotides, TAT, okay, TAT, these are heterogeneous bases. That's the 10 sequence. Then the 35 sequence usually consists of TTGA, CA. So you should take note of these nitrogenous bases. Then you should differentiate them from the eukaryotic promoters. Now you see that eukaryotic promoters, they are extremely diverse and difficult to characterize. And main contain the Tata box. Now get the difference. There's a difference between the Tata box and the TAT, okay, which is in the prokaryotes. So this side it's Tata, then this side it's TAT, okay, with the double A there. Okay, so the Tata box, we can call it the Tata, like triple A at the end, which in turn binds the Tata binding protein, which assists in the formation of RNA polymerase transcriptional complex. The Tata box lies very close to the transcriptional start site, often within 50 bases, So it is important to take note of the difference of the in terms of the promoters, okay, that are actually found in eukaryotic cells and prokaryotic cells. Okay, so unless there is a question on concerning transcription, we are going to call it. Is it a night or an evening?
Yeah, I've got a question on
Yes.
exons and introns.
Yes.
Which enzyme is responsible for the joining of the exons?
So that's ligation. So you expect ligase enzyme to ligate. Yes. Yeah, to form those covalent bonds. Yes.
Right.
Yeah. Is there any other question? If there's no other question, we'll meet up tomorrow. Tomorrow is Friday, isn't it? Yeah, we'll meet up tomorrow for the continuation of our sessions.
And so I don't know if you can find a way of sending these notes.
Or we'll discuss that with the organizer. Yeah.
All right.
Yes. Okay. Otherwise, you guys will have a good evening.